chapter. So chapter 9, as I know, has been omitted from the CBSE syllabus, right? So after chapter 8, it's chapter 10 that we will do first. Anyone here who's studying from some other board than CBSE? All of you are from CBSE, Rifat, Aisha, Donna, and Hanifa?
Yes.
Okay, very well. CBSE, right?
Yeah.
Okay, cool. Okay, so let's begin with this new chapter, which is microbes in human welfare. Okay, cool. So as the name suggests, let's start from the very basics, like we always do. In the previous chapter, which is chapter 8, where we studied human health and diseases, one of the major factor that cause diseases is pathogens. And these are microbes that we studied about: bacteria, virus, protozoa, fungi, worms, etc. So these small organisms are not very good, right? They cause a lot of diseases in humans. So we studied the bad aspect, where they can be pathogenic and where they can be harmful. So in today's class, let's also look at the other side of the coin. So there's always two sides to anything, right? So all microbes are not bad. There are microbes which are good for us, or which we can utilize for our welfare, for human welfare. So before we begin, anyone know about any good microbes from day-to-day life, from around you? Any good microbes you are aware of? Are all microbes bad all the time?
No.
Then which are the good ones?
Probiotics.
Sorry?
Lactobacillus, I guess. Hello?
Yes, Aisha.
For the milk and all.
Perfect. So one example that Aisha gave, which is directly from your syllabus, is Lactobacillus, correct?
Yeah.
What does Lactobacillus do, Aisha?
Okay, I have to explain it.
No, just in one sentence, tell me that what it is used for. Like you can say it is used for making houses.
Yeah, like for curd and all, and even in milk also, right?
In milk, it doesn't help.
Basically it helps to digest. Digest, right?
Digest what? We have
Uh,
acid in our stomach to digest things for us.
Okay, 1 minute.
Your first sentence was correct. It helps in curdling of milk. So we make curd from milk. Okay, and you must have seen whenever you want to make curd from milk, we add the inoculum, which is curd only, right?
Yeah.
We put a little bit of curd in the milk, and then the next day the whole milk is curd. So that curd contains lactobacillus. Good. Anything else? Any other example, anyone? Apart from lactobacillus, which you have encountered in your day-to-day life? Microbes can be used for a lot of things. Good microbes. Just lactobacillus. Sorry? Yes. Come again.
Probiotics.
Your voice is not very clear, Hanifa. Can you speak again? Okay, you're writing probiotics. Okay. Fair enough. So Hanifa said probiotics. What are probiotics? You also have to tell it. Probiotics. What is they used for? What is the benefit? Where is the welfare?
They are like they're bacteria, but they're like the good bacteria which like keep us healthy.
So where are they? How do they keep us healthy? They must be, yes, you're right, probacteria for good health, but where are they in our body?
They're um, they're like I'm not sure about that. I know they fight off the other bacteria to keep us healthy.
Yeah, they're present in our intestines. Okay. We have a whole colony of many bacteria in our intestine, which is called a gut microbe, gut microbiome, or gut microbiota. And that's very important for us because they synthesize very important biomolecules for us, including some very essential vitamins like vitamin B12 complex. So those are our probiotics, which stay in our gut, in the intestine, and they are called gut bacteria. Anything else?
Is
Yes, Aisha.
Bifidobacteria a good bacteria? Sorry, good
Which which one? Which one?
Bifidobacteria.
Your voice is breaking, Aisha. I don't know if it's my network or yours. Is my voice also breaking for you people?
No, sir.
No,
That's
okay.
only.
Okay, then it's probably
Is it
your.
okay now?
Yes.
Sir, Bifidobacteria is a good microbe.
The moment you start taking
Hello?
the name of the bacteria, I lose your voice. You will say everything and I will understand it. Hello, is my voice audible? But the moment you will say the name of the bacteria, I cannot hear you.
Okay.
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 Okay, you typed bifidobacteria. I've not. What does bifidobacteria do? You have to also tell me that because it's something very specific. I'm not aware of this bacteria.
Hello, sir.
Yes, tell me.
Sir, can you repeat again?
Yeah, I'm saying that you have to also tell me what is bifidobacteria. What does it do?
Yeah, I know about it.
Because I don't know about it.
I know, like gas system, like basically for acid
think
and maybe some acidity gas problems and all. I think so.
Okay.
I'm not sure, sir.
So if it is a part of the probiotics only, so it's already covered. So bifidobacteria is, I think, a part of probiotics. Like the way you, according to what you are saying, if it helps with the stomach and intestinal regulation and maintenance, it is a part of the probiotics, the gut bacteria only. So it comes in this category. So no one of you, I'm talking about microbes, so you don't have to just stick to bacteria. Bacteria are not just, there are many microbes apart from bacteria which are useful to us. How many of you like, yes?
I'm not sure of the name, but we use certain microbes to ferment things.
Perfect. What are those microbes? You're amazing.
I'm not sure what it's called though.
But you know to which phylum or to which class, category of organisms they belong?
Not sure, sir.
Not sure.
Oh,
Okay, have you? Yes, correct. So what you're talking about is yeast, right?
Yes, sir.
It's also known as baker's yeast. And the scientific name is Saccharomyces cerevisiae. So this is used for fermentation. And with this we make bread, your cakes, all are made because of fermentation. That's why you see, so during fermentation, CO2 is released as a gas. And that's where you see that in the dough, which is fermenting to make a bread, a loaf of bread, you will see these small, small holes. So the bread is like spongy in structure. Am I right?
Sure, even baking powder also, right?
Yes, it contains like baking powder is different. Like you're talking about baking soda and baking powder, there's baking yeast. So those are they also perform they chemically perform this, but as organisms, yeast do fermentation biologically in the cells. Okay, cool. So yeast is one. So to add, anyone would like to add any other microbe which is useful to humans or useful to you around yourself?
So is it like, are they in cosmetics and beauty products?
Cosmetics. I don't know, like no experience, zero experience. So you tell me. Someone from you, tell me. Do we have microbes in cosmetic products? I thought these are neutral, sometimes harmful, but mostly not very useful chemicals, right? They can be neutral, not doing anything. But I'm not sure about microbes in cosmetics. Okay, so Lubna has joined. Lubna, is this your first class?
Yes, sir, it's my first class.
Okay, welcome to Learnivio. My name is Rahul Kushwaha, and welcome to biology classes. I'm one of the faculties of biology at Learnivio. So we have just started with microbes in human welfare chapter. And Lubna, would you like to introduce yourself, a quick introduction, so that we know where are you from, what do you wish to do with biology as a subject, how do you like the subjects? This will help me to design a better plan of teaching if something specific is required for you.
My name is Lubna and I'm from Bangalore. And
Okay.
well, for biology, well, I want to go for MBBS, so I chose this as the mainstream. So
Yes, that's compulsory then, right? Good.
yes,
So you want to become a doctor, you want to appear for NEET, right?
yes.
Very good, a very noble pursuit. I wish you all the best, and I'll try to help you the best I can. So I am Rahul, and by profession, I am a biology researcher. I'm pursuing my career in biological research, currently doing a PhD in neuroscience and learning, memory, and behavior. And I like teaching, so I like preparing students for competitive entrances to make a career in biology, research, medicine, anything associated with biology. So it's just like I have a thing for teaching. So welcome to my classes. I hope you will learn as well as you will enjoy. So we have just started. So please, so there are two rules, very simple rules. One, be discussive. You have to discuss and discuss. Okay. Second, do not accept anything which I say just because I'm a teacher. Teachers can be wrong. Teachers are wrong at many times, and there's no shame in admitting it. But I'll try to be factually correct as much as I can. But you have all the rights to ask for explanations, proofs of anything that we are teaching or discussing in the class. Okay, Lubna?
Okay, thank you, sir.
Okay, great. Yes, so and anything you would like to add to? We are talking about any good microbes. So Aisha said Lactobacillus is one good microbe because it's used for curdling of milk. Then probiotics were named. Who named probiotics? Was it Hanifa? Yeah. And then yeast was by Donna. So anything you think is a good microbe and is around us? Because we have studied about a lot of bad microbes in disease chapter, human health and disease. So showing some better side.
I don't have any idea right now, sir. I'm not sure.
Yeah, because the most common ones have been taken, so it's not your okay. We also have Shaheen. Shaheen, is this is your first class as well?
Yes, sir.
Oh, I'm so sorry. I got confused you with someone else named, I think it's Shaheen or something similar. So would you like to introduce yourself just like Lubna quickly? Do you happen to know Aisha by any chance?
Yeah, I know her.
Me, Lubna, Shine, we know each other very well.
Oh,
And
that's great.
well
So
as along
you,
with Mariam, so
okay, so you're classmates.
yeah.
Right.
No, yeah, me and Shine
Cool.
are class.
Okay, so Shine, would you like to introduce yourself?
My name is Shaheen. I'm from Bihar.
Where are you from
And
in Bihar? Because I am also from Bihar natively.
Bihar, I'm from Muzaffarpur.
Oh, great. Okay, and how do you like the subject biology and what do you wish to do with the subject?
Well, I think MBBS.
MBBS. Okay, same as Lubna. Good. So
Yes.
you have already heard the rules. So, and I think you have
Yes, sir.
started well already. Okay, cool. So let's keep going. Anything else? No one has an idea because all the common ones are taken. So let me tell you very quickly. If you don't know the microbes name, but still there are many things. For example, we make cheese because of microbes. Cheese is possible because of microbes. Otherwise, we would not have cheese. Apart from cheese, there are medicines for which microbes are used. I'll give you an example of penicillin. So penicillin is made from Penicillium notatum, which is a fungi. We also have microbes for other bioactive chemicals or bioactive molecules, which are used in treating many diseased conditions and to help with many symptoms. Then we'll come to industrial uses of microbes, and towards the end in this chapter, you have to learn about microbes in sewage treatment. So how do we also use microbes to clean the sewage water?
In beer also,
Yeah.
microbes are also used, right?
Where? In
beer and wine.
Yes, in alcohol. So that's again fermentation. So there are many microbes that can be used to make alcohol of different kinds and different concentrations. Mostly it's yeast, Saccharomyces cerevisiae is also used, and other microbes can also be used which can ferment. Okay, so let's begin. So how we will divide this chapter is we'll talk about microbes we'll start from the house, the most common one, what we use in house. Then we'll go to, yes, Atif.
Sir, can you say diatoms are good microbe?
Diatoms, you have to just prove that what is their benefit to us. The
Yes,
good
sir,
means they should have some benefit to humankind. Yes, tell me.
Yes, sir. They like produce diatomaceous earth, which has like a lot of uses. Like for example,
example?
it is used to remove like unwanted material from drinking water. Then it is also used for like filtration of oils, syrups, polishing, etc.
Yes, very good. A very valid point, and thank you for bringing it up. But if you notice, the product that we are using, which is diatomaceous earth, it is not the living diatom itself. It is not the microbe, but the remains of microbe. When these microbes die, diatomaceous earth is the it's the accommodation, or I should say, millions of their shells stuck together and hardened as stone. So that is diatomaceous earth, right? Because even when diatoms die, their shell, which is made up of silica, it's not destructible, so it does not destroy, so it stays like that. So that's where we get this diatomaceous earth. It's like a stone. So in real terms, we are not using the microbe here, but what is remains from the microbe. That's why we will not study diatoms in this chapter. But your point is very valid. Do you understand, Atif? Because we are not utilizing them alive as microbes, but it's a microbe product. For example, petroleum, all petroleum product, majority of them have come from the bacteria. From millions of years, these bacteria, which has been buried, they are the major source along with plants and animals. But since it is the product and not the microbe itself, will not count that. Okay.
Yes.
So for example, bioactive molecules that we are synthesizing is also product. But to make this, we are using those molecules in present. right? Not it's different from how we are using diatomaceous earth. Make sense to you? Atif, does it make sense to you?
Yes.
Okay, cool. So how we'll proceed is, let's do this. We'll start from household products like microbes which are present in our house. Then we'll go to industries, and finally we'll go to sewage treatment, which is like on a city level. Then we'll go to science, where we produce biogas from microbes. We produce biocontrol agents, medicines, and fertilizers. Okay, so starting with the first tier where good microbes can be utilized. Microbes in household products. So already Aisha started with one such microbe, which is Lactobacillus, right?
Yes, sir.
So in household, we know the first kind of bacteria is lactic acid bacteria, also called lab. And one such bacteria is Lactobacillus. What does the word bacillus tells us about? You have studied about bacterial cells and their shapes, right?
Yes.
So what is bacillus telling us?
Lactobacillus bacteria.
No, bacillus. What is bacillus? You must have heard name like coccus, spirillum, vibrio,
They're
bacillus.
rod-shaped.
Yes. So bacillus means rod shaped, and lacto means they use lactose and they produce lactic acid. So they are lactose dependent bacteria and they are rod shaped. So if we just do this word lactose dependent and rod shaped. Okay. Now what do lactobacillus do? There are other bacteria along with lactobacillus also, which come under the broader category of lactic acid bacteria. But taking an example of lactobacillus, what do they do? How do they? So what is the function they serve? Curdling of milk. This we know. But how? How does it happen? Any idea? So lactobacillus produces lactic acid. Okay. Now lactic acid is an organic acid, just like other acids, citric acid, formic acid. So lactic acid is acidic. So as these bacteria keep growing in the milk, they produce lactic acid. Milk becomes acidic, and under the acidic condition, milk slowly starts coagulating. So these bacteria digest the milk proteins and make the environment acidic, because of which in acidic environment proteins coagulate. They come together and starts binding to each other. That's where milk slowly and eventually starts coming together and become curd. Is it clear, everyone?
Yes, sir.
Yes, sir.
Yeah. And if you have seen, if I put so always whenever you have to make curd, we keep it at an optimum temperature. Too cold or too hot, the curd will be proper curd will not form, right? If it is too cold, if you keep the milk in a refrigerator at 4 degrees after putting the inoculum, it will not become curd very well, or it will slow down because the bacterial growth and replication will slow down. If it is too hot, again, it will make the curd bad. So it needs an optimum temperature at which these bacteria can grow properly in number and make curd out of milk. And we always add this inoculum. You must have seen adding inoculum to the milk. Inoculum is nothing but curd itself in small quantity, because we have to introduce this bacteria, the lactobacillus, in the milk. So a drop of inoculum already contains millions of these bacteria, depending on the volume of milk that we are converting into curd. So apart from just curdling, is there any other benefit of curdling the milk, making the milk into curd, or is it just because we like curd?
It's homemade. Like
It's
if we do it in the home.
even if it's not
If it
homemade, if you buy it from the
do it
market,
in
curd
the
is curd. So in the market also they make curd like on an industrial scale. They also make curd with the same process. They add inoculum and make the milk become curd. There's no other process. But what is the benefit of making curd from milk?
Nutritional values increase.
What is the benefit of making curd from milk? No benefit, right?
The
There is
nutritional value increases.
Yes. So curd has increased nutritional value. Increased nutritional value. Okay. And how?
I have one, but I don't know I should say or not. But
Yeah, you should say.
it's for digestion.
It's good for digestion, yes, because you know why it's good for digestion. It's good for your gut actually, not for the digestion. It's good for your total gut because Lactobacillus is a good bacteria. You need it in your gut. If it goes in your gut, it will do good things. And plus, I was talking about vitamin B12, right? So in a curd, there is increased level of vitamin B12, which is not present in the milk. Okay.
Yes, sir.
Very good. Now, so this is one lactic acid producing lactic acid bacteria which are used in household products. Second, also we studied kind of. Can anyone tell me? Cheese. So, how many of you have watched Tom and Jerry?
I have watched.
Okay. So let's go to cheese making bacteria. Okay, now these are not. So if you have watched Tom and Jerry, you must be aware of what I'm going to draw now. Can anyone tell me what is this?
Cheese.
It's a slice of cheese, right?
Yeah.
Yeah.
Yes.
So what are these things? What is this thing? These whole kind of things. This kind of cheese specifically is called Swiss cheese with these big holes. How do these holes come up? Someone sits and cuts the hole in this cheese. You have identified it just because of these holes, right? Otherwise, you could have said it's a pizza slice or it's a paneer slice or anything else that can be sliced. But you all said cheese because you saw, oh, this we have seen. If I use a yellow color.
They're called eyes.
Sorry?
The holes in the cheese, they're called eyes.
They're called eyes. You can call them eyes, nose, ears, whatever, but where do they come from is my question.
From milk, right? Like
From milk. Cheese is made from milk. So when we were making dahi or curd, these holes were not there. We were using a bacteria. Now we are using another bacteria. So this bacteria is known as Propopioni Bacterium Sharmani. Very fancy name I find for a bacteria. Propopioni Bacterium Sharmani. So this is used to make Swiss cheese, and how it makes Swiss cheese is because this bacterium kind of puts flavor through fermentation, and these big holes are due to release of CO2 in large quantity when this cheese is being made. So there is bacteria present in this cheese all around, and these bacteria produce lots and lots of CO2, which comes out from that milk as these and forming these big holes. Okay. Similarly, so this is called Swiss cheese. Remember that there is another kind of cheese known as, for this we use a bacteria. There is another kind of cheese which is called Rockford cheese. QUO, I'm not wrong. Rockford cheese. This has flavor, so it's flavored, and this flavor comes from a fungi. You allow a fungi to grow on cheese, and again same fermentation happens, so it becomes cheese, plus this time you also have a flavor, particular flavor, which comes because of these fungi that grow on the cheese. So these are all good bacteria, because ultimately we are ending up eating them, putting them in our mouth and getting them in the system. So they are clearly not bad bacteria. Are these three clear to you? Swiss cheese, Rockford cheese. The difference is Rockford cheese is made through a fungi. Fort and fungi have F. Swiss cheese is from a bacteria. Okay, Sharmani makes Swiss, fungi makes Fort. Will you remember this, people? This can be asked.
Yes, sir.
So for cheese and for curd, normally, very commonly in our houses, we use microbes. Now, going on an industrial scale, let's also talk about some things that we can do in home also, but we can also do in industrial levels. So microbes in industrial use or industrial products. Yes, what are the things that we make in industries? So in industries, if we need to ferment something, we don't do it in vessels, we do it in big small, we don't do it in small pots or small vessels like we do it in home, but instead we do it in very big container vessels called fermenters. So all the fermented beverages. So first thing is, in industries, this Saccharomyces. And you know what is Saccharomyces?
Yes.
It's fungi, right? So Saccharomyces cerevisiae is used for fermented beverages. Can you name some fermented beverages produced on a commercial scale?
Alcohol.
Alcohol.
Beer.
Wine.
Yes, alcohol, beer, whiskey, brandy, rum. All these come apart from this.
Yeah.
Nothing else? Anything else?
Champagne.
Champagne is again alcohol. Okay, so
Wine.
alcohol is of two types. Let me tell you. Wine is also alcohol, no? So either it is distilled or it is non-distilled. Okay. Now your homework is that you will. I think you can easily find it in NCERT. If not given, just do some internet search and figure out which alcohol, whatever you name, like champagne, wine, brandy, rum, beer, whiskey. I heard all of these. Which one are distilled ones and which one are non-distilled one, and what is the difference? Okay, so first A, we use it for fermented beverages. We also use it for bread making. So bread is a big industrial business, all sorts of bread, brown, white, bun, everything. So there also we use Saccharomyces cerevisiae, and same, the concept of fermentation is used, but this time not in a liquid but in a dough. Okay, in a dough the fermentation happens. You know what is fermentation, people?
So is it something to do with the anaerobic respiration?
You have studied it in your junior classes, the process of fermentation.
Sir, you were stuck.
Sorry,
Sir, you were stuck. Now it's fine.
was I stuck for others as well?
Yes, sir.
Yes, sir.
Okay, I'm sorry. Okay, one moment. Is my voice better now? Let me keep the video off for some time to get the bandwidth for the voice. That's more important. Yes, so I was asking, what is fermentation?
It's the process in 1 minute, sir. I know it.
Yep. Yeah, take 1 minute.
Yeah, it's a process by which molecules like glucose they break down
Mhm.
and anaerobically, I guess. I studied somewhere with this.
Yeah, you're correct. Fermentation is a metabolic process or a chemical process, biochemical process by which molecules such as you can use molecules like glucose, which are broken down anaerobically. If you break down the same molecule, the glucose, aerobically, it's not called fermentation. What it is called then? What do we all do?
Respiration.
Respiration, right? We call them respiration. And each and every cell of our body does respiration. Yes or no? Aerobic
Yes,
breakdown
sir.
of glucose. And what is the byproduct that comes out through respiration when we do the aerobic breakdown of glucose?
CO2.
Okay. And how is it different from fermentation? What are the byproducts that come out in fermentation? In fermentation also CO2 comes out, right? That's where we are having these holes in the cheese and these spongy structure in the bread. But what is the difference? Anaerobic breakdown cannot completely break down all the glucose into CO2. Some intermediate.
So with oxygen also,
Sorry,
along
come
with,
again, Aisha.
like it also comes along with oxygen, like not completely with CO2.
No, oxygen is not there, no. If there was oxygen, then it would have been an aerobic respiration. It is an anaerobic respiration. So how can oxygen be there? How can oxygen come out?
Like before that, I don't know how to see it.
If oxygen was present or if oxygen was produced. Your voice is breaking, Aisha. Would you like to come on a stable network or rejoin? Is it breaking for everyone, right?
Yes.
Yeah.
Yes, sir.
Yes, Aisha, your voice is breaking a lot. So try coming on a stable network. Yes, but so fermentation is anaerobic breakdown of glucose, and in which the glucose is not completely broken down into CO2, and there are some intermediate products left. Like sometimes when anaerobic respiration happens in our muscles, you know that if you are vigorously playing or running or doing exercise or gymming, the first day you start getting pain in your muscles. You know why is that pain there?
Due to
Like
production of
there's no
lactic acid.
enough oxygen.
Yes, Shaheen, correct. If there is no enough oxygen, sorry, I'll come to you, Atif. Then what happens, Shaheen, if there is no enough oxygen?
If there's no enough oxygen, the pain starts.
What? How? You just started the journey and went to the credits. When there is no enough oxygen, pain starts. End of the movie.
Uh,
So if I hold my nose for 5 minutes, will pain start in my muscles if I don't get oxygen in my body?
no,
Then something else is happening, right?
like
When we are
not
running,
enough supply of
Breathing.
oxygen.
yeah, so what is happening when enough oxygen is not there? I just told you. You just have to repeat that. Yes, Aisha.
Lack of breathing. Am I audible now?
Yes, you're audible. Lack of breathing. Yeah, you can say less breathing. So when we are running or vigorously playing, we do not breathe in, though our breathing rate increases, but we are not able to breathe in enough oxygen that is required by the muscles, which are vigorously utilizing oxygen as a fuel. So what happens? Oxygen become deficient, and from aerobic respiration, which is complete breakdown of glucose, the muscles shift to anaerobic respiration, which is incomplete breakdown of glucose, and as a result, lactic acid is produced in the muscles. And lactic acid is an acid, it's an organic acid, so that creates pain and cramps in the muscle. So acid, when it comes in contact with muscle, it hurts, right? It pains. So if acid gets deposited in the muscles, your legs, your body will start paining. After some time, if you keep drinking water properly, that acid is slowly and slowly again taken up by the cells, sent for metabolic breakdown, and then broken down. So it's flushed out, cleared from the system. So after some days, you feel normal. Okay, so one is Saccharomyces, which is used in fermented beverages, which comes under industrial products. Anything else? So you have to figure out, write down, this is your homework. Distilled versus
Even
non-distilled.
shampoo and conditioners is also there.
Shampoo.
Yeah.
Shampoo have microbes?
Yeah, that head and shoulder and all like dandruff free.
Which microbes are there in shampoo?
Wait. I think
Do you
so.
have a shampoo bottle right now in your home? Do you have a shampoo bottle accessible to you right now, Aisha? Okay, your voice is breaking. I don't know what you are saying, but if you have, just look at it, read the ingredients, and tell me if there is any microbe in it. Okay.
Yeah.
So apart, yeah. So go and read and tell me which microbe is there. Okay. By that time, let's move on from Saccharomyces or from like fermented beverages to the second industrially produced things where we use microbes. They're called antibiotics, right? You all have heard of antibiotics? Not just heard, we all have taken antibiotics. Cool. So Aisha has a cat, apparently. By this time, all of you know, right? Just don't tell her. So she has a very cute cat who loves to study biology with her. I was just telling that you have a cute cat and you keep yelling at your cat sometimes. So it's everyone should just be okay with that. Did you check which microbe is there in your shampoo, Aisha?
I was just reading.
Yes. If you find any microbe, do let me know. Okay. I don't want to just give you a.
I don't think there are microbes in shampoo.
Come on, Lubna. So Lubna just gave the spoiler, I just wanted Aisha to do some digging. Yes, Lubna is right. There are no microbes in shampoo.
Because if there are microbes, then it's basically the shampoo is spoiling and it can cause health problems.
But for dandruff, it's there. I read somewhere, like in Pinterest or somewhere, like for the dandruff and all.
So what is dandruff? Why do we need microbes for dandruff? If you just know what is dandruff, you can just. What is dandruff? Dandruff is nothing but your own scalp getting dry and flaking off from your head. Dandruff is not something different. It's our own head scalp. So if our scalp gets dried, it flakes off and become dandruff, small white thing substances. So if you can use something which can keep your scalp wet and oily, you will not have dandruff. Simple. So you can just do that by chemicals, or you can just do that by oiling. You don't need microbes for that. I know people put all kind of funny things, starting with egg to dahi to putting a Nemo in something and paneer and whatnot. I don't know if they help or they do not, but they might be helping. That's why people are doing it. There is something called beer shampoo, which has beer, and probably that's why Aisha is saying that there are microbes in shampoo. But if you just read the ingredients of the shampoo, there is no active microbe in the shampoo. Though there are certain products which can be synthesized from microbes, but not microbes themselves. Okay, Aisha? So Lubna is correct. Okay, so coming to antibiotics, we all have taken antibiotics. Can you name any antibiotics? What does the word antibiotic mean, first of all, quickly? The word anti means against. Anticlimatic. You have not heard the word anti anywhere?
Sir, can you repeat again what you asked?
I asked, what do we mean by anti? Is my voice still lagging, people? It should not lag now.
No, sir.
It's for me, actually, I think so.
Yeah, I think Aisha, it's your network.
Yeah.
Because my voice is okay for others.
Antibiotic means disinfectant.
Anti means against, and the word biotics comes from the word biota. So antibiotic means against life. Now my question is,
bacteria, I guess.
Yeah, whose life it is against? Our life?
No.
Then?
Bacteria.
The microbe's life, right?
Mm-hmm.
So antibiotics can be of different types. Normally in your textbook, the word antibiotic is used for antibacterial drugs. If they are antiviral, they are against viruses. If they are antifungal, they are against fungi. They are all antibiotics because they are against some or the other kind of life. But the most common antibiotic we will be talking about is penicillin. You have heard of penicillin?
Yes, sir.
Yes, sir.
It is an antibacterial thing. And where does it comes from? Where was penicillin first discovered? Whom? By whom? This name you should know, Alexander Fleming. Alexander Fleming was working on a bacterial strain, and the bacteria he was working on was Staphylococcus. Now the name coccus, what does it tell you?
Spherical.
The shape.
Just give me 1 second, people. There's someone at my apartment door. I'll just be back in 30 seconds, okay? Yep. Hello, am I audible?
Yes, sir.
Yes.
Thank you so much. Yes. So what does it tell? What does the word Staphylococcus tells us about? So Alexander Fleming was working on Staphylococcus. So he was maintaining plate cultures. So the way bacteria are maintained is there is a plate on which you have a agar medium, the medium which provides food to the bacteria, and you have bacterial colonies growing on it. So this is how we maintain the bacteria. So this bacterial colony was Staphylococcus, on which Alexander Fleming was working. He was a microbiologist doing some research. Now what happened? One day he left some plates around near the window because he was not going to use it. Might be they were some failed experiment or something, but there were bacteria growing in that plate. So he thought, okay, later on I'll just get rid of these bacteria. He left it there. After a few days, so there are many kind of versions of this story, that he went on a holiday, he came back, or something like that. But what happened is, after some time passed, and when he came back from the holiday, or from just normally one day, he came back and saw that there is something growing on these plates. So there is a mold-like structure growing at one side of the plate, and because of this mold, in the vicinity of this mold, all these bacteria, they are now dead. So no bacterial colony is living. All the bacterial colony are dead. But on the other side of the plate, there are living bacterial colonies. So he thought that maybe this reddish substance, this mold, is secreting something that's killing these bacteria. And that is how he first identified penicillin, because this mold was a fungi whose name was Penicillium notatum. So from Penicillium notatum, the name penicillin comes, because he identified and extracted the substance which could kill bacteria in its vicinity. Now he figured out that Penicillium notatum, which is a fungi, can give rise to or secretes penicillin. But there were two more scientists apart from Alexander Fleming, which is first. Second was Ernest Chain. And third was Howard Florey. Second and third. So these two, what they did, they for the first time established penicillin as an antibiotic, as a medicine. Established it, okay, made it effective antibiotic. So they produced effective antibiotic. So Fleming discovered, and they gave meaning to his discovery, application to his discovery. Produced effective antibiotic from penicillin. And they together, all three of them, were awarded the Nobel Prize in 1945, just after the Second World War ended, because penicillin saved many lives in the Second World War of many soldiers who were wounded and had undergone bacterial infections on the field. So they were the easiest targets to test this drug on, because anyway, soldiers who are wounded and who have got bacterial infection from the soil and from water or from anywhere in their wounds, they are anyways about to die or get their legs and hands amputated. So it's better that you give them some drug and try to see if it treats them, and it did. So antibiotic worked, and these soldiers were saved, and it was a big help for America. And this discovery was awarded Nobel Prize in 1945. Okay. Is it clear, everyone? So after penicillin, penicillin is not the only antibiotic. We have many other antibiotics. If you go and figure out, we have kanamycin, we have strepto something, I'm forgetting the name. There are many antibiotics now, and their derivatives which are in the market and in use. So other organisms or other microbes also produce certain substances which kill bacteria. Okay, so today we cannot imagine a world without antibiotics, because if there were no antibiotics, even the most common bacterial infections would have claimed lives. People would have died because of infections that cannot be treated or controlled. Now, today, whenever there is a bacterial infection, we can just control it. If it is on the skin, you put a lotion of an ectopic antibiotic like sulfamycin, etc. And if it is internal, you take amoxicillin or penicillin or streptavidin. All these tablets you take, these are antibiotics, and you get healed. But earlier it was not the case. So millions of lives have been saved around the globe every year because of antibiotics. But there is a problem that's growing. You know what is the problem that's growing nowadays with antibiotics? Anyone has
The
heard
bacteria,
of? Yes, Atif. Yes, please.
the bacteria will develop resistance after a while.
What does it mean? You're correct, but would you like to just simplify it a little bit for your batchmates?
Like when we consume the antibiotic more and more often, as like time progresses, the bacteria will undergo evolution to resist against the antibiotic, so that it will no longer be effective.
Okay, so basically there are many mechanisms of antibiotic resistance, but what Atif said broadly holds true. So earlier, when antibiotics were discovered, or as a new antibiotic is discovered, first bacteria gets completely wiped off by that antibiotic. But over the time, bacterial strains and bacterial populations develop resistance against the drug. So we cannot kill these kill these bacterial cells that effectively with the antibiotic. We either need a higher, more powerful dose or multiple doses to kill the same bacteria that 20, 30 years ago could have been just killed with a very low dose. Now, can you think why is it happening? How is it happening? Because remember, bacteria can only reproduce through asexual reproduction. So technically all bacteria must be clone of each other, right? And if they are clones of each other, then how are they developing resistance? Anyone? Any weird idea in your head? So there are
The
some.
mutations.
Perfect. One is mutation. Very good. But with mutation, only a few bacteria can get mutation, and though it's true that they replicate very fast, in every 20 minutes or 30 minutes, bacteria like E. coli can replicate and divide into 2 cells. So that's true. Mutation is one. Very good. Apart from that, there is something that bacteria show, which is not a part of this chapter, but since we are studying antibiotics, I'm giving you as a homework in this green box. You can go up and read about horizontal gene transfer in bacteria. Okay, so bacteria has this property. Some bacteria, not all, some bacteria can just pick up random DNA fragments from their environment and can put it inside their DNA. So if this random piece of DNA contains anything which is useful, like a like a antibiotic resistance factor, then this bacteria will get antibiotic resistant, and then through the same process of horizontal gene transfer, which is called transformation and conjugation, they can pass on these genes to their neighboring bacteria, not to their offsprings. If transfer of genes are happening from parents to offsprings, then it is called vertical gene transfer, because it's going down the line, right? Can you understand? This is called vertical gene transfer from one generation to another. Horizontal gene transfer is where in the same generation, that gene is getting transferred to many individuals in that population. And mostly horizontal gene transfer is very more common in unicellular organisms, specifically these bacteria. So this is one way, but they get resistance, and this resistance is creating a big problem now, because in the coming time, we are afraid that bacteria which are getting resistant to multiple drugs, they are called multi-drug resistant bacteria, MDRS, multi-drug resistance strains. So MDRS bacteria pose a bigger threat to our health system. And you know where are these MDRS bacteria found in the most number? You'll be surprised to know. Where you have the highest chances to get this MDRS infection. Can you think where?
Pond.
Sorry, ponds. Okay.
No.
Then lakes, rivers, polluted water bodies, right?
Yes, sir.
Sewers. No. The highest chances of you getting infected with MDRS is hospitals. The very place where you go to get treated is becoming the hub of MDRS infections because of the use of antibiotics there. So antibiotics are readily used and microbes are being exposed to a lot of antibiotics. So if you take, that's why it is said that if you are taking antibiotics, complete the dose. Don't leave in between. Because what you are doing when you are leaving the dose in between is you are exposing your system and the bacteria in that system, the pathogens in the system, to antibiotics, but you are not exposing them enough so that they get killed. You are just exposing them and leaving them. So which means you are showing them the weapon through which we can kill them, but you are not killing them. So next time they will learn, next time when they develop this resistance, the reason that they survived is because they developed resistance. And if these resistant bacterial strains continue to live, the next time they will infect you, all of them will be drug resistant strains, and it will be difficult to treat it with the same antibiotic with which you could have treated it earlier. So this is the problem that's happening. In the last 50, 60 years, we have misused and overused antibiotics to a great extent. And that's one reason that antibiotic resistance is developed in bacteria. Is it clear, everyone?
Yes, sir.
Yes.
Okay, so this was just for you to understand the concept. So to make antibiotics and to make fermented beverages, we use it in industries. What else can we use it for in industries? What else do we make in very large quantities in industries? We make chemicals for our use, we make enzymes, and we make bioactive molecules. Right? All these things we make in industries, and then they are sold to pharmaceutical companies or to other companies which use it to make something else. Now, talking about commercial and industrial production of certain chemicals, first obvious candidate is organic acid producing bacteria. Can you tell me what is an organic acid and what is an inorganic acid? Some chemistry 101. What are organic acids?
Organic acids are the biochemical compound.
Sorry, come again. You have to be a little louder because one, your voices are breaking. Second, if you are very, very low, then I won't be able to understand it.
So
Come again, quick.
organic
Yeah.
is something which is not dangerous as it can be used by us.
What does the word organic means? Dangerous or not dangerous, we'll talk about it later.
Organic compounds. Okay.
Organic, like extracted from nature, something like that.
Exactly. Something which is synthesized inside living systems is organic molecule. So there are many microbes that produce these acids. So if they produce these acids, mostly they produce it as a product of any metabolic activity or as a defense mechanism. You know, when honeybees sting, what is present in that sting of honeybee that makes you
Acid.
what acid?
I think formic acid, I'm not sure.
Yes, it's called formic acid, also present in some ant stings and honeybee stings. So it is synthesized in living systems, but is it harmful to you or not?
No.
So then why is being stinging you? Why are you in pain when bee stings you? It is to an extent, right? It irritates your cells. It causes swelling, infection. Sometimes it also causes fever in some people. So it's a defense mechanism of the bee. Acid anywhere in more than it is required or when it accumulates is harmful. That's why all the acids that are produced in our own body, they never stay as acidic in their acidic form. Let's say there is citric acid, CH3COOH. This is acetic acid, right? Chemistry, this is the formula for acetic acid.
Yes.
Let's make the structure. There is a carbon with 3 hydrogen, CH3. Then there is another carbon, C, with a double bond O and a OH, right?
Yes, sir.
So inside a living
Yes, sir.
system, this acetic acid will never stay as an acetic acid. It will stay as acetate. It will donate this hydrogen and become CH3COO negative and H plus will be released. So this part where this hydrogen goes away and this part remains is called acetate. All the acids in our system stays as acetate form. Okay, acetate, aspartate, you know. Do you understand?
Yes, sir.
Do you understand, people? Citrate,
Yes, sir. Yes.
lactate, etc. Okay.
Yes, sir.
Yeah. So acid producing bacteria or acid producing microbes. I should not write bacteria. I should write microbes here because there are some fungi as well. So organic acid producing microbes. So first is what produces acetic acid. What is the use of acetic acid? Why it is commercially produced? Anyone knows acetic acid? What is acetic acid? How many of you love Chinese food? Chow mein. To cook chow mein, there is something used which is known as vinegar. Also to make pickles, something is used known as vinegar. Right?
Yes, sir.
What is vinegar?
Mostly acetic acid.
Yeah, it's not mostly, it's a diluted acetic acid, but it is acetic acid. Okay. It's around
Yes, sir.
10% acetic acid. Yeah. Okay, so acetic acid is produced by a bacterium called Acetobacter aceti. So a common name, very easy to remember, Acetobacter, the bacteria that produce acetic acid, and the species name is aceti. So Acetobacter aceti is a bacteria. Yes, Atif.
Yes, sir. What is the difference between white vinegar and normal vinegar?
Sorry, come again.
What is the difference between normal vinegar and white vinegar?
normal vinegar and white vinegar. Vinegar, acetic acid is colorless normally. So vinegar is 10% vinegar is white, I think. But I think when you put vinegar on a flame or you cook it, it changes color. Kind of. I'm not sure though. You have to check. I think it's just commercially different types of vinegar, either in terms of flavor or something is added to it. Because vinegar in its pure form is colorless only. So if the colorless one is called white vinegar, then the other one must be having something changed. Okay, can you figure it out, Atif? I'm not very sure what is, because
Sir, what about the apple cider vinegar? Does it have any difference in?
yes, the origin, the difference is in the origin. As it says apple cider. What does apple cider means? You know apple cider?
No, sir.
Apple, simple.
The-
What do you think is an apple cider? It is made from fermented apple juice. So it's, yes. So if you are making it from apple, it's called apple cider vinegar. Okay.
Oh.
So you crush, basically what you do is you crush apples, you know, stampede on apple and crush it under big crushing things. And the juice that comes out, which if you just slightly ferment it, it becomes epiphyse kind of a drink. Have you tasted
Yes.
partially fermented apple drinks, epiphyse kind of things? If you completely ferment it, it will become apple cider vinegar. Okay.
Thank you, sir.
Yeah, that's what I know. But about the white and what was the other one, Atif? White and
Never mind,
black.
sir. White vinegar is normal vinegar.
Sorry.
Sir, I asked like what is the difference between white vinegar and normal vinegar, but actually white vinegar is normal vinegar. Both are the same thing.
Okay, great. So what is the abnormal vinegar? Is there anything which is not white but a vinegar? I think apple cider vinegar has some color and flavor.
Yes, sir.
Yeah, but yeah.
Even jamun vinegar is also there, right? My mom, she got it from India.
Which vinegar? Sorry.
Jamun.
Jamun.
Yeah.
That the fruit jamun. Wow.
Yeah.
I have no idea about. I literally have no idea about these fancy vinegars. I'm very sorry
My mom,
about
she
it.
got it from India.
But yeah, I'm honestly I'm hearing it for the first time. Jamun vinegar. Apple cider vinegar is common, and vinegar is of course common in cooking. But jamun and what other? These are some good if they exist. But they are vinegar, you know that. And a bacteria that can produce vinegar for you, sorry, that can produce acetic acid for you, from which you can later on make vinegar, is Acetobacter aceti. It is used in large quantities in industries to make this acid. Another acid, organic acid, which is useful to us is citric acid. We know that citric acid is normally produced by citrus fruits, right?
Yeah. Even
All the
that Mirinda, Limca,
I don't think Mirinda and Limca, any of these two has citric acid in it. I think they all are
Orange
carb.
and lime.
Sorry?
Orange and lime.
Yes, all the fruits have citric acid, all the fruits belonging to the citrus family, orange, mosambi, lime, lemon, all these contain citric acid. And I'll tell you, as a kid, I was very naughty, and I think most kids are that way. So if you take the orange pulp and squeeze it from the outside, so the pulp of the orange contains this citric acid, and if it goes in someone's eyes, then the person is sure will cry and has to clean the eye with water. So it's not harmful to the eye in any way, but it's very irritating and causes irritation and pain. So that's citric acid. But it can be commercially synthesized on a large scale using bacteria which are called, sorry, a fungus which is called Aspergillus. Aspergillus niger. This is a fungi and it produces citric acid. Okay. Then one another is very common that we just studied about, lactic acid. What produces lactic acid?
Lactobacillus.
Yes. Lactobacillus is an example. Lactobacillus bacilli. It's again a bacterium. And butyric acid is also used commercially. Butyric acid is another organic acid, and it is produced from Clostridium. Clostridium butylicum. That's why it's called butyric acid. It's again a bacteria. Okay. These are some common organic acids that are used. So chemicals contain this. You already know that for ethanol, ethanol is used as what? What is the use of ethanol apart from making alcoholic beverages? We all have been through COVID phases. What is the one thing that you used to keep close to your heart more than anything throughout the COVID?
Mobile phones and gadgets.
Gadgets?
No, like some gadgets do contain ethanol or something like that.
Sanitizers. Gadgets don't
Sorry.
contain ethanol.
Yeah.
Your hand sanitizers. Hand sanitizers are 70% ethanol. That's how they dehydrate and kill, desiccate the COVID virus. So we all used to rub our hands with sanitizers every now and then. It contains alcohol, 70% alcohol. Also, your medicines, many medicines which are tablets, especially. So these tablets are basically, the drugs are produced as powders. Then these powders are as a solvent. The liquid which is used is not water to make tablets. In water, bacterial contamination can be there. So ethanol is used as the solvent to make many medicines, including your cough syrups. Cough syrups has alcohol. So alcohol has many useful properties that are utilized, one of which is antibacterial property or antimicrobial property. Microbes do not grow in alcohol. So it is used as a solvent in many pharmaceutical medicine producing companies. Also as hand sanitizers and cleaning agents. And have you seen, any one of you have got an injection? Before the injection, they clean that surface, rub cotton. The cotton is dipped in something and is rubbed. Right? Even before surgeries, the body is cleaned. They are all done by alcohol-based spirits. Okay, so for alcohol, we use yeast on a very large quantity to make alcohol. And alcohol can then be used for sanitizers, beverages, and medicines. Is it clear?
Yes, sir.
Everyone? Okay. Cool. So let's stop here for today. And in the next class, I'll be talking about some more drugs and products, bioactive molecules. So we still have to talk about enzymes and some bioactive molecules that we will talk about in tomorrow's, not tomorrow, the next class. And then we'll come to sewage treatment plants. That is one thing you have to study in detail because it is asked in boards as well. So that we will do in the next class. So I'll stop here. We still have 3 minutes. So I'll stop here and I'll take questions. If you have any questions, you can go through every note. You can ask if you have any doubt.
Sir, any example for butyric acid?
Sorry, any example for butyric? Like example of the organism that produces butyric acid, or where is butyric acid used?
The use.
Okay, so uses of butyric acid is mostly in let me think, where can we use butyric acid? It is used as esters. There are many. Now I have to explain you what is esters. paints. Paint is one good example. You know, paints and coatings that are done to make something resistant to degradation or rust,
Yes,
or
sir.
spray paints. In that you use butyric acid. Okay, that is one place. Another benefit is butyric acid is also taken, like it's given to people who are suffering from um, obesity. Like it helps in weight loss. It is also anti-inflammatory. Okay, and some researchers also suggest that butyric acid is also used to make anti-cancer drugs. Okay,
Yes, sir. Thank you, sir.
so it's helpful in some regulated amount in food. Okay, and in prebiotics, these Yakult and things, there is some amount of butyric acid also added. Okay, and commercially it is used for paints, and sometimes it's also used for your, so you what do they call it, yogurts. You know, yogurts, there's a difference between curd and yogurt, you know that. So yogurts also, some yogurts have some butyric acid in it. Okay,
Yes.
Yes.
any other doubt, people? Anyone has any other doubt? Okay, if no doubt, then I'll see you all in the next class, where we'll try to go towards the, so this is a short chapter. I think it will not take more than 2 classes. Sewage treatment plant is the longest part that we have to study, along with biogas production, which both of which I will take together, and then we'll biofertilizers, and I think, what is it, BT cotton. Yes, these, some few of these topics are important, so we'll do this in the next class. Okay, chalo, see you then in the next class. Bye bye.
Thank you, sir.
Thank you, sir.
Thank you.